Okay, this is where we left off, slide 36. Sorry I went a little slow today, but we can get caught up here. Only about 10 more slides, so it shouldn't take too long. Just wanted to reiterate, here we use this Teflon insert to isolate this neuron into chamber B, A, and C with the soma and cell bodies in A. And you can see down here that same sort of thing where you see dendrites out here in B and C and the soma in well A. And there's nerve growth factor in all of these. And then when we remove the nerve growth factor, it's clear that the nerve growth factor is the chemical signal that is allowing the axons to grow to their appropriate target, as we see in well C. And when we take it out and don't have nerve growth factor in well B, then we lose those connections. We lose those dendrites. Interestingly, nerve growth factor not necessary to keep the cell alive because the somas don't die, the cell bodies don't die. So nerve growth factor in this case is a guidance signal for the connectivity of this neuron, not for its survival. And another one of these neurotrophins, maybe BDNF, is necessary to keep the cells alive. And without BDNF, the cell would not be in the right place, so it would die. Okay, so obviously if we're recognizing these different neurotrophins like NGF, nerve growth factor, BDNF, neurotrophic factor 4, 5, or 3, then these chemicals have to be binding in some way or being recognized in some way by the cells, and they do so through these receptors that are much like metabotropic receptors. They're called the TREC family of receptors, and when they bind, they release enzymes inside the neuron that then keep the neuron happy and healthy and tell it that it's in the right place. Neurotrophins also, in addition to cell survival and migration, cause differentiation. So here we see a stem cell, a neural progenitor cell. And then if that stem cell is exposed to 4 different types of chemicals as it migrates to its destination, those 4 different chemicals cause that stem cell to become a different type of cell. So leukocyte-inducing factor causes that cell to become a sensory neuron, right? If we have glucocorticoids, it's going to cause that cell to become a chromatophin cell progenitor, and then glucocorticoids cause a further change into a chromatophin cell in the stress system. So often we might see 2 steps. So here we see a step where we have 2 different chemicals. So FGF2 causes this stem cell to become a sympathetic nervous system progenitor. And then if the cell is exposed to NGF, it becomes an adrenal cell, one that's going to release adrenaline. However, if it becomes exposed to ciliary neurotrophic factor, it becomes an ACH or cholinergic cell. So here we have a multi-step process where we turn it into sort of a common cell, and then for the sympathetic nervous system, and then the specific neural type. All right, apoptosis. Excuse me. If we end up in the wrong place, the cell is going to, and how do we know we're not in the right place? Because there's not the right neurotrophic support. Then a cell is going to undergo apoptosis. And it does so through a programmed cell death. So this programmed cell death involves increased calcium, and when there's too much calcium inside the neuron, it releases a protein called the Diablo protein, which we know that has to be bad just by the name of it. And the Diablo protein, shown here, then disables the inhibitors of apoptosis. So basically, we have a check and a balance system. So normally, cells are prevented or protected from undergoing this programmed cell death by the presence of these inhibitors of apoptosis proteins. And so when Diablo gets released, because there's too much calcium, it can disable that, and then it activates that program of cell death. In normal development, depending on which area of the brain we're in, different areas have different rates of apoptosis. We can lose 20%, which is 1 in 5 neurons, which is pretty big if you think about it, all the way up to 80%, where we're losing 4 out of 5 neurons through programmed cell death. So that's why we have overproliferation of neurons. We're making way more neurons than we're going to end up keeping, and this allows for neurons to make mistakes and then undergo apoptosis, but it also allows for the other neurons to have a greater chance of forming the right connections and ending up in the appropriate places. Finally, after we have those 4 major stages, overproliferation, right, migration, differentiation, and apoptosis, then we get myelination. And again, we have different myelin for the central nervous system. We have our oligodendrocytes that are going to wrap around and form these branches of myelin sheaths. And then Schwann cells in the peripheral nervous system, where we have a single sheath wrapping around a cell. Synaptogenesis, the creation of new synapses, is similar to overproliferation in the fact that cells are going to connect to more things initially than they're going to stay connected to. So we want to overproduce synapses and then prune back to the synapses that are functional. And that, again, allows for us to account for inappropriate synapses and it not hurt the overall population of correct synapses. Synapses are one of the main primary developmental things that's happening in childhood. So we talked about by 1 year of age, a brain, 1-year-old's brain looks a lot like an adult's brain, and that's in terms of proliferation of neurons and apoptosis. But synaptogenesis is where we're getting better connectivity in the brain, and this is what is allowing the child to develop intellectually, emotionally, from a personality standpoint, all of that. And so here we see neurons in the cortex of a newborn, okay? And what we see is that the number of neurons doesn't dramatically change, but what dramatically changes by 2 years old is the connectivity of those neurons. So, in the first year, we have lots of overproduction of synapses, and then from 8 months to early adolescence, 11 or so, we're getting a pruning, and so much of the learning how to walk, learning how to talk, is happening through pruning of inappropriate synapses and stabilizing the appropriate connections to allow motor function, to allow the coordinated movement necessary for speech. And then you'll see that that pretty much plateaus, and then we do lose synapses later in life, and again, that's part of the aging process. So this process of pruning, we overconnect. So here we see 3 axons coming in to 3 targets, and all 3 axons are connecting initially with all 3 targets. In this case, there's 1 correct target for each of these, and it's the support of the activity and neurotrophins that allows the pruning to the appropriate connections. And so now we have 1, 2, 3 axons hooking up with 1, 2, 3 targets, and the other connections have been pruned because they did not receive the right activity or the right neurotrophic support. Right? When we talk about the motor system, coming up in the next section, exam section, we'll talk about the fact that some muscles, the pruning results in a 1-to-1 muscle fiber ratio. So we have 1 axon connecting with 1 muscle fiber. And this gives us the best control over our muscles, whereas here we have axons coming in and connecting to at least 2 fibers. So we can't individually move those. We have to move them together, right? And so we don't have as much control. Now, this is something that is what we call plasticity, and we'll talk more about plasticity on Thursday. But this plasticity is the ability to change connections. And so depending on your use of your muscles, you can actually change the fiber-muscle ratio, and you can learn to make more controlled, finer motor movements. For example, when you first start learning to play violin or guitar, you're kind of clumsy with your fingers, and you can't physically make your fingers move very fast or move to the right places in the right increments. But with use, practice, a lot of practice, you become much better at that. One thing that's changing is in your brain, you're memorizing the motor patterns and learning the motor patterns, but in your peripheral motor system, you're also decreasing the number of synapses, and so you're gaining more finite, better motor control there. All right, and finally, the role of experience. So we've talked about how experience can drive genetic expression, and experience drives development, synaptic connectivity in particular. And so here on the left side, we have a neuron from a fish that was raised in a tank by itself. Here we have on the right a neuron from a fish that was reared with other fish. And just being in the presence of other fish is a more enriching environment. In fact, being in isolation is a deprived environment. And we see the effect on the synaptic connections. Many more dendrites, many more little synapses all along this neuron than from the one raised by itself. So, the key in terms of experience driving brain development is that when we have deprivation, we get negative effects. If you never hear music, ever, in development, then that could impair your ability to be able to discern musical notes and hear music in a way that other people hear as an adult. If you never ever hear it, right? But playing lots and lots of classical music for a baby is going to produce adequate experience, but it's not going to make them more easily become a classical musician or have some extra special abilities. So stimulating environments may be beneficial, but you can't go in the opposite direction of deprivation to become super at a task because you've been over-enriched. Experience also affects the maintenance on neural connections. So if you learn to play guitar and you get better at all those patterns and then you stop for 5 years, when you pick the guitar back up, right, there's going to be a loss of those connections, and it's going to take more reuse to regain that ability, to regain that. All right. Rett syndrome is a very rare condition. It is sex-linked, and it happens in girls more than boys. So 1 out of 10,000 births show Rett syndrome. It's a common cause of severe mental retardation. We think that it also occurs in boys, but is such a bad thing in males that they probably spontaneously abort in utero, and so it's actually probably a lethal condition, and we don't get very many live births of boys that have Rett syndrome. And the reason is because it's a dominant mutation on the X chromosome, and boys only have one X chromosome. They have an X and a Y. So if they have this dominant mutation, it's much more powerful in boys than in girls, where they have a second chromosome that may not have that dominant mutation on it. So they essentially have one broken and one normal chromosome. And so it doesn't kill the girls, but it does produce this severe mental retardation. And what does it do? Well, it impacts dendritic branching. So here we see neurons from the brain, from an autopsy brain of a girl that had Rett syndrome. and we see much, much fewer connections to the point that it becomes difficult to cognitively function and compared to a non-REDS brain. All right, so connectivity in the brain, very, very important. Does development end? Well, we talked a lot about stem cells, undifferentiated cells that can be built into any type of cells, the basic building blocks, right? And they're found in the ventricles of the brain. And we know that we can find stem cells in adult brains. So if we have stem cells, it doesn't make sense that we're not in some way producing new neurons. We do know that stem cells can migrate to the olfactory bulb and become new neurons in the olfactory bulb. Particularly if it becomes damaged, we can regain function in that particular pathway. And we've also found some evidence of new neurons in the hippocampus. And what's the hippocampus involved with? Learning and memory, yes. So as we continue to learn and form new memories throughout our lifespan, that might be an area that would be very important to be able to generate new neurons. So while I said that CNS neurons are sort of permanent and when they get damaged you can't replace them, in general, yes, that's true. Does it mean that we don't ever get new neurons in adult brains? No, we probably do get some new neurons in adult brains, but we don't get enough in the appropriate areas to compensate for when we have neuronal loss. All right. So there is some debate over whether this happens in humans. It's obviously very difficult to study because people don't like for you to do a brain biopsy and take out some of their hippocampus and see if there are new neurons in it. So everything has to be done post-mortem, and that's more difficult than in animal models. So, but it's likely that this is happening in human brains as well. And certainly, development doesn't end in terms of synaptogenesis and forming new synapses, and primarily in how we learn new tasks. All right, so here are your review points for the first half of the lecture and for the second half of the lecture. On Thursday, we're going to talk about CNS damage and neuroplasticity. It's very important. It's the last part before the exam. If you have questions, you can email me. I'll send you an answer back, and I'll also post those to the middle page. I have a website linked on there where students have asked other questions in the past, so you can see what students have asked about before. I'll just caution you that if you are reading the questions from years prior, and they say on slide 3 of the neural signaling lecture, what does something mean? That's probably not the same slide 3 that you have, because I change my lectures every year. So you kind of have to ignore slide numbers and that sort of thing, but you can see my answers to their questions as well. All right, that's it. I hope you have a great rest of your Tuesday. I will see you in lab on Wednesday.